Okay, so now basically, guys, when we talk about this enhancement in food production, the first thing, what is this chapter really going to talk about? This is going to talk about, like, first of all, what is your plant breeding? What are the different ways in which we can bring about plant breeding? What is the actual experiment that has to be done? Then it is going to talk about, like, when we talk about, like, different hybrids. Okay, what are the different types of hybrids that we have produced so that, we can get good yields or good varieties. Then we talk about, like, rice, we talk about wheat, we talk about your sugarcane. Okay, so these are like different varieties we talk about. Finally, after talking about this, we start talking about what is your different kinds of diseases, what are the pest resistant plants. Then we talked about, like, how we can improve the quality of food, okay, or improve the quality of plant. Okay, whether it is going to be by biofortification or whether it is going to be by normal fortification, something of that sort. And finally, towards the end of the chapter, we are going to start talking about your genetically modified organisms. Okay, so how we can produce these genetically modified organisms, and then we have what is tissue culture and how do we go about this tissue culture. That is what we need to know in this chapter. Okay, so guys, the first thing, okay, so first thing we're going to start off with is what is your plant breeding exactly. So let's try to see this thing. Okay, so guys, when we talk about this plant breeding is nothing but it is a branch of your botany. Okay, so as we're talking about plants, so it is going to be a branch of botany, and this branch of botany basically it deals with the collective use of art. Okay, it deals with collective use of art and science for changing the hereditary, for changing the heredity of plants, changing the heredity in order to create desired varieties or desired types. Okay, so basically, in short, what do we mean by plant breeding? Plant breeding is basically you are actually looking at some character. Let's say we are talking, we are talking about, like, good flower production. Okay, so here in this case, you are going to target only that, good flower production capability of a plant, and you are going to bring about a reproduction of that plant in such a way that only that good flower producing capability is going to be preserved. Rest other things we are not concerned with, but we are looking at only that specific thing. So what are we trying to do? We are trying to change the heredity, or we are trying to change the genetics of the plant in order to create your own required types, or in order to create something that you really want. That is nothing but your plant breeding. But now, whenever we talk about plant breeding, okay, is there any application of plant breeding? Why should you actually choose some kind of a plant? Why should you actually, choose some kind of a character? Yes, there are many applications. Okay, uh, many applications, because see, sometimes what happens, some kind of plants, they are going to be more economically viable. Sometimes in some cases you are going to see that some plants, they are suited best to some kind of an environment. There are going to be certain plants in which, you are going to see excessive demand in the market. So basically, you are going to go for this plant breeding for these kinds of purposes. Fine. But now, how can this plant breeding be brought about? Is there any way that we can do this plant breeding? Okay, yes, there is a way how we can do this plant breeding. So basically, when we talk about this plant breeding, this can be brought about by various things. Like, so first of all, we can make use of principles of genetics. Okay, so basically, guys, I told you, Mendelian genetics. What are we going to go for? We are going to select some specific character and we are going to focus only on that character. That is what Mendel's homozygosity. So this is going to be done on the basis of your principles of genetics. Now, if you're not going onto this, then you can use some other techniques as well. So second thing, you can look out for its taxonomy. You can look out for its hierarchy, where it lies in which family, how does it look like, what are the specific characters, or you can look at its physiology, or there it can be some kind of pathology, or there can be your requirement of agriculture, or we can also do this thing, we can do this artificially with the help of using recombinant dna technology. Recombinant dna technology, that is nothing but your plant breeding. So we can do all of this by doing this. Okay, now always remember, why are we going to go for this plant breeding, guys? Because there are certain characters which are going to be really really important. Okay, for example, like let's say, we are using this recombinant dna technology, and we are going to make a plant. Okay, make a plant well suited for the environment. So let's try to see what are the uses and what is the importance of doing this thing in a plant. Okay, with this, okay, with plant breeding, we can achieve. Okay, so first thing, guys, what we can do is we can increase their genetic pattern, or we can alter their genetic pattern. Okay, so we can alter their genetic pattern, and because of this genetic pattern, maybe we are increasing its demand in the, I mean, we are supplying the demand in the market, so we alter its value, or we increase its value and its use. So for use, we call it as utility. So for example, if we look at, can you read the first statement sentence? It is a branch of botany which deals with the collective use of art and science for changing the heredity of plants in order to create desired types. Okay. Okay. So now, apart from this, guys, what are we going to use it for? Okay. So the second thing that you're going to see is, so guys, when we are going to do this thing like plant breeding, what is the actual aim? Now, guys, whether it is plant breeding or whether it is your animal breeding, the entire idea is to increase the production. Okay, so that is the main aim. That is why we are like selecting, like, some specific breeds or some specific kinds of plants or some specific kind of animals. Okay, so here the next thing is we are going to increase the yield. Okay, so to increase the yield of the product. Okay, or maybe to improve the quality, increase the quality of the plant, make it like, high yielding or high producing capacity, or maybe enhance its character, or maybe this is the one we can increase its nutritional values. Okay, so maybe it's nutritional values or nutritional qualities, or maybe we are increasing its tolerance to environmental stress. For example, okay, when I talk about environmental stress, what can this be? Environmental stress meaning if you are having high amounts of your like temperature, okay, extremely high temperatures, extremely high salinity, extreme dryness. Okay, so for all these things, they are going to be made more tolerant. So environmental stress. Okay, apart from that, guys, by using this technology, like by using plant breeding, we can create pest resistant plants. Same way, we can have pest resistant, we have insect resistant pest plants, or insect resistant plants. Okay, and along with that, guys, we can also make it like more sustainable. Okay, sustainable, or okay, like more adapted to the environment. Even these kinds of plants can be produced by making use of your plant breeding techniques. Okay, but now see, no matter what, okay, so we are trying to do these things, guys. Always remember, why are we doing this? So that we get plants which are going to be of better quality, we are going to get plants which are going to be of high nutritional qualities, so that they are going to keep on keeping giving the same characteristics again and again, again and again and again and again in the next generations. So guys, what happens is, when you are going to do this kind of a technique, the seeds and all that are going to be produced, those are also going to be of much superior quality. The chemicals and the pesticides, the fertilizers, whatever that are going to be required, they are going to be less if you're like breeding this in the most proper manner. Okay, and plus these are the plants who are going to produce high yields. So what is the benefit of it? You are going to get more amounts of your product, you are going to get more amounts of money by selling the product. That is why we are going to go for this kind of thing. Okay, so this is nothing but plant breeding. But on the same line, so we are going to have your animal breeding. Now animal breeding, we have always already talked about, like animal husbandry. So basically animal breeding is nothing but you are going to breed animals to increase their yield, or to product, to increase their yield. Basically it can be either your wool, it can be your milk, it can be any kind of other product. So animal breeding is generally done to increase the yield of the product. Okay, but now in animal breeding, the first thing that we have to talk about is domestication. So, guys, what is domestication? See, animal breeding, it is a little difficult unless and until we do not domesticate the animals. Okay, so what is this domestication, guys? Basically, it is a general term. It is not like pertaining only and only to your animals. It belongs to your plants as well. What is domestication? Domestication is nothing but it is the process of bringing. It is the process of bringing plant or animal species under human management. Under human management. And that is nothing but your domestication. Okay, but now, guys, let's try to see what really is going to happen in the time of your plant breeding techniques. Like what are we really looking at? What exactly is going to happen? How are we going to go about? So guys, this is very, very, very simple. Okay, because what are we going to talk about? We are going to talk about only this thing, that is your Mendel's experiment only, but in a little modified manner. Okay, so guys, when did we actually go for all this plant breeding techniques and all? Okay, guys, when there was this green revolution. Okay, so who's the father of green revolution? Father of green revolution is your Norman Borlaug. Okay, we'll be talking about him in a little while. And in India, the same green revolution, this was brought about by Dr. M.S. Swaminathan. Okay, so his role was especially in India only. What he did, how he did, we will be seeing it in a while. But then, what are the techniques that these people followed? These people followed your plant breeding techniques only. Okay, so first, let's try to see, guys, what are the different techniques? What are the different things that we need to bring about? Okay, so 1 minute. There's a question over here. Uh, Sir, I have seen that sometimes two highly achieving race horses are also forced to produce their offspring so that the offspring also becomes a race horse. Is this the same thing? Yes, absolutely right. This is nothing but your pedigree. Okay. And always remember, especially if you are dealing into horses and all, horses, they run in lakhs and lakhs. Okay. They are like really expensive. Okay. But then what they will always see, whether the offspring is from a good family. In fact, if you know, in dogs also, we believe we have the same thing. We always look at the parents, because the product, that is the offspring, is going to be a product of the parents. So you have to see if the parents are good, then definitely the good genes are going to come in the offspring. So whatever you're saying is absolutely right, Shruti. Absolutely right. Okay, so now we are going to go for your plant breeding. Okay, but plant breeding, now you're going to see that there are different steps in this. Okay, and why will we go for plant breeding? Like I said, guys, we are going to be in need of some good varieties. Okay, we need to have some better yielding varieties, and that is why we will go for plant breeding. So, first thing is in this, we are going to go for hybridization. Okay, so let's try to see what is hybridization. How does it really work? Oh, 1 minute. There's one more question. Their pedigrees do really matter. Yes, yes, absolutely. It's very, very important because whether it is a show line breed, whether it is a working line breed, all of that information you can immediately come to know by looking at the pedigree. So basically, how it is like, for every organism there is a registration committee. Okay, so for dogs it is the Kennel Club of India. Okay, so you have to register your pet out over there. So what happens is when you register his parents and all, they are also registered out over there. Hopefully they are registered. You get an entire family tree. This is your parent, these are your grandparents, these are your great grandparents, these are your great 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 grandparents. And then you can find out whose characteristics are being like transferred from one generation to the another. It is that way. So it is very important. Okay, very important. And people pay like crazy for like these kinds of like properly verified pedigree pets. Okay, there's a huge market for it. So animal breeders and all, they will especially target pedigree this thing only. Okay, pedigree animals, not the normal ones and all. Okay, so now guys, we are going to go into your plant breeding. Okay, and we are going to see what are the different steps included in plant breeding. Now, guys, this question, this question used to be asked for 7 marks in the earlier version, but for you guys, this will be asked for 4 marks. Okay, so what are the plant breeding techniques? And you should also be able to you should write down what are the different steps. Okay, so in plant breeding, guys, what are we especially going to look at? We are going to be looking at your hybridization. Once again, guys, we have done hybridization many a times. What is hybridization? Hybridization is nothing but mixing of two individuals to get a better offspring who is going to have combined characteristics. That is nothing but your hybridization. Okay, so this is nothing but what is hybridization? This is nothing but combining characteristics of two good parents. That is nothing but hybridization. Okay, and what is the importance of this? With while you do this hybridization, guys, the offsprings that we are going to get. Now I said, see, they are going to contain characteristics of both. So these offsprings, whatever that we get, okay, these are going to be better yielding. Offsprings are going to be the best. They contain both the characteristics, and these we say they are going to show hybrid vigor. So they either show hybrid vigor, that means they are going to be better than their parents, and plus they are going to have both the characteristics, or we also call this thing as heterosis. So that way. Okay, so these are the ones they are going to have. Yes, Sarah, you wish to ask something? Hello, Sarah. Hello, hello. Sarah, do you wish to ask anything? Hello.
Hello, sorry. I'm sorry.
Okay, okay, no problem, no problem. Okay, so guys, this is what we are going to exploit. Okay, we are going to exploit the property of hybrid vigor, meaning whenever you have two good parents, okay, you are going to try to take the parents and you're going to try to mix their characteristics. But guys, is it always that you are going to have offsprings with best characteristics? Is it always going to be that way? Quickly answer, guys. So let's say, okay, I am having two plants. Okay, so let's say one produces big flowers. Okay, it has big flowers, and but these flowers have a very short lifespan. So 1 day lifespan. Okay, and then there is another plant who is going to have very small flowers, very small flowers, but now this has a lifespan of 10 days. Okay, this is hypothetical. Okay, but understand. So guys, what are we going to look at? Okay, we are always going to see that, oh, I want to combine these two characters. Okay, so I want big flowers and I want to combine 10 days. Yes, very nice, Himanshu, very nice. Okay, even Komal has answered it rightly. Okay, now guys, whenever we do this thing, always remember, you are not going to always get the characteristics of what you want. There can be chances that everything may go the other way around as well. So how do we know? So guys, let's imagine using now guys, you tell me, when I do a cross of this thing, okay, big flowers and 1 day lifespan, small flowers and 10-day lifespan, I am going to cross these two plants. What am I going to get? Once again, guys, which law are we going to follow out over here, guys? Can anybody tell me which law of Mendel are we going to follow where the characteristics are going to get exchanged, meaning you are going to see recombination taking place? What is that character? Yes, absolutely right. Parnavi has given me the right answer. What is the law we are seeing? We are looking at, yes, very nice, Shruti. We are looking at Mendel's third law, independent assortment of genes. So, guys, when we do this cross, what are we going to get? We are going to get big flowers. 1 day, this is going to be the original parent. Second one, we are going to have small flowers and 10 days. This is going to be the other one. But guys, what are we going to be looking for? We are looking for hybrid vigor. So you are also going to see this big flowers and 10 days. And the other one, this can also go with this. You are going to have small flowers and 1 day. Now, guys, now see, this is not like easy. Like I am saying, ki, Ore bapre, chalo, you take the plants, you just mix them and you just sow them, and you have got the plant of your own choice. No, 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 no. It is not that simple, guys. There is a process. So now, what are you going to get? See, these, when you are going to get, these are nothing but your seeds, guys. Okay, these are going to be the seeds obtained from the fruits. You have to take this, you have to sow them. After you sow them, they are going to grow and they are going to start producing flowers. Now it is your duty to actually look out for them and see that, oh, if this is really going to last for 1 day or whether this is going to last for your 10 days. Then you are going to select this. Now guys, once you have selected this, once again, your job is not done. You have to verify. What are you going to verify? You are going to verify that, oh, whether this is really going to stay like alive for 10 days, or whether this is like just a chance character. You have to undergo self pollination. And guys, quickly tell me, self pollination up to a period of how many generations? Guys, quickly, 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 quickly. Very nice, Neha Singh. Very nice. It is going to be for a minimum period of 3 generations. And then you will come to know that, yes, you have got hybrid vigor. You have got the entire plan that you were looking for. So, guys, this is how it is going to be done. Okay. Fine. Everybody has got an idea how we are going to go about this thing. So remember, there is a process. Okay. And this is a little tedious process. It is not an easy process. You have an offspring and immediately you see everything. No. Okay. Shruti says yes. Okay. Is this the same thing that Einstein was asked by that his smartness and someone beauty can be combined? And he answered, yes, it can be other way around as well. Yes, absolutely. It is exactly the same thing. Okay. Yeah. Chal. Okay, so I hope everybody has understood what we are trying to do out over here. Okay, so I will, 1 minute, I will just remove this part because this is not really needed, but I wanted to explain you out what happens. Okay, but now, guys, there can be different types of hybridization. So, see, just like, how we talked about, your breeding, we have inbreeding, we have because it is not there for you. This is just to make you understand. So that is why I put it in another file. This is not a part of how it is. That is why. Okay. So now, guys, when we talk about this, okay, when we talk about your hybridization can be of different types, guys. Okay, just like how we have talked about your like when we talked about your breeding, we have inbreeding, we have your outbreeding, crossbreeding, interspecific hybridization, outcrossing, right? So the same way we have hybridization in plants as well. Now how is it going to be? Now we have three different types. Okay, so let's try to see what are these three different types. And guys, keep this thing in mind. This is very, very, very important. Okay, because why? You are going to see that they ask you this thing in your NEET exam. Okay, not that important from the board's point of view, but very important from the NEET point. So, guys, these hybridization they can be intravarietal. Okay, so first is intravarietal. Second is intervarietal. Okay, and the third one, guys, this is going to be intergeneric. Okay, so let me just quickly explain it out to you. How is this thing going to be? So, guys, always remember. Okay, now always remember, intra meaning what? Intra meaning it is going to be within the same. Okay, inter meaning within different. So for example, once again, guys, intra school competition, intra school meaning it is going to be held within your school only, that is intra school. Inter school meaning there are several hundreds of schools they are participating, that is inter school. So intra and inter, first get this thing clear. Second thing, guys, when we talk about your intravarietal, okay, so when we talk about your intravarietal, what is really going to happen? This is going to be between the same plant, okay, meaning the different varieties of the same plant. Okay, so different varieties of the same plant, meaning there can be like the same plant, its offsprings, they are going to have like different kinds of characteristics. Okay, so within the same intravarietal. Okay, second thing, if we are talking about intervarietal, then it is going to be between different plants, but they have to be of the same species. Okay, so two varieties between two varieties of the same species, then you can see that they can be belonging to the different parents, different plants, okay, different species, different locations, maybe geographical locations, maybe that way. So between two varieties of same species. Okay, then after that we have your intergeneric, meaning this can be within the same genus. Okay, within this family of this, within the same genus basically. 1 minute. Sorry. Okay, so between this is going to be between uh, two varieties. Okay, but this is going to be between two varieties of two genera. Genera meaning your this thing, two genera of the same family. Okay, then guys, there is one more out over here which I forgot to put it down. Intergeneric, and there is interspecific as well. Interspecific is the one which is between two organisms of the same genus. Okay, interspecific, this is between two varieties of the same genes. That is going to be your interspecific. So guys, these are going to be like, these are going to be four different varieties by which we can bring about hybridization, selecting like, what you are actually looking for. Guys, always remember Mendel's homozygosity. What is it going to result into? It is ultimately someday or the other, it is going to result into your inbreeding depression. And in order to overcome inbreeding depression, you will either have to go for intervarietal, intergeneric, or interspecific. Okay, so this is what you need to know. Guys, always keep it in mind, intergeneric, so it has to be between two genera. Okay, interspecific, it has to be between the same species of the same genus. Okay, so just keep this thing in mind. Okay, but now guys, sometimes what happens, okay, now this is a little different. Now sometimes you may know that two plants, they are going to be like related, but then they are not like, immediately related, they are going to be distantly related. Like, one may be like, great 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 grandfather of some generation, and the other generation is more modified and it is seen in some other place. Okay, so now you can also bring about crosses between the two. So when we have crosses, okay, crosses between distantly related plant, crosses between distantly related plants, okay, then this is known as wide cross, or this is also known as your distant cross. Okay, guys, this is important from your need point of view. Okay, so they will ask you, what is this? If you have a like a relative, so they will generally not use this distant family member, they'll use a relative. Okay, if a cross is done with a relative of the plant in consideration, then what kind of a cross is this? Okay, fine. Sure. But now, guys, now we are going to move into the actual process of hybridization. What are the different steps involved, and what are we really looking at? Okay, so guys, the first step for hybridization, okay, the steps in hybridization, what are these? So the first step is you are going to look out for variability. So we say the first step is collection of variability. Okay, okay, one minute, there is one question. I did not understand what is a distant cross. Okay, fine. So let's imagine you have the same parents, let's say the same family. So let's say we have the grandfather. Again, we are talking about this thing, okay, like with plants. Okay, so understand this thing. Okay, so we have grandfather, then you have father. Okay, let's say, but this your father also has a brother, he is going to be your uncle, right? Now let's say the father has a son and he has a daughter. Let's imagine this thing. Okay, we are just talking about, like plant examples only. Okay, and then let's imagine this uncle also produces like a son and he has a daughter. Now in your plants, okay guys, remember this is plants. Okay, in your normal animals like humans, we cannot do this thing. Okay, but now if this son has to be mated with this daughter, then aren't they like related to one another? But they are not like really, here I'm showing it really close, but then there can be like, different pedigrees. When you do a cross like this between different this thing, like, different people but from the same family, they are going to have this common ancestry. So I can say they are related to one another, not maybe like, closely related, but maybe like really this thing, like, distantly related. So for example, say we have Komal Patni out over here, right? So we have Komal Patni. We also have this actress called Disha Patni. Now they may be closely related, or they may be, distantly related. Who knows? Because Patni, the surname remains the same. Everyone getting this thing? So if there is a cross between those two people, like Disha Patni, and maybe we have Komal Patni's brother, maybe. Okay, in that case, what are you going to get? You are going to get a distant cross, because you are not immediate family members. You are going to be distant related. वो मेरे आंटी के आंटी का cousin का लड़का है. So he is related to me, but distantly related. I hope you got this thing. Yeah. Okay. So that is exactly actually what we are talking about. And I hope everybody got this thing as well. Same mother or different, it doesn't matter. It has to come from the same lineage. It has to come from same lineage. But if you are from the same mother, then it is not going to be a distant cross. Then you are going to be like, mating with your sibling. So that is going to be like, inbreeding kind of thing. Okay, so it is going to be, it has to be a little different. Like, uncle ki uncle ka, ya mere cousin uncle ke ladke ka ladka. It is going to be something of that sort, distant relative of the family. Okay. So guys, when we go for this, hybridization, the first thing is we are going to go for your variability. Okay, so what do I mean by variability? Collection of variability. So here in this thing, guys, you are going to make sure, okay, you are going to make sure that all the different alleles, they are going to be present. So you are going to have an entire collection of different kinds of genome which is going to be present around. Meaning what? Let's say you are talking about a rose plant. So you are going to collect all the variable possibles, all the possible variations of the rose plant. Meaning you are going to see some plants, they are going to be more fragrant. You are going to have some plants who are going to be producing large flowers, some plants who are going to be producing flowers over a period of great time. You are going to see plants which are going to have like genes which may be like giving the flower the ability to survive for like 10 days. Maybe the other one maybe have the ability to produce less thorns. Maybe another one is going to be pest resistant. Another one is going to be like fertilizer free, like it is like it is rich in your nutrients. So you are going to collect all the different possible combinations out over there. That is called as your variability. Okay, so you will all possible, okay, all possible characters and traits of the plant. Okay, like for example your rose plant, all the different characters, okay, you are going to select. Okay, they are going to be selected and they are going to be preserved. Okay, so the first thing is that. But now, what is this entire collection called? This entire collection of all the various alleles, okay, entire collection of alleles, this is called as germplasm, or we also call this thing as the germ pool. Entire collection of different types of alleles. This is known as germplasm, or we also call this thing as germ pool. So guys, just understand this thing. This is nothing but a collection of plants where all the possible characters and their genes are available. So tomorrow if you want to combine two characteristics from two different plants, you have both the plants in your hand. It is that way. Okay, so guys, understand this thing. Now this, because we have like collected this thing, we can also call this thing as germplasm collection. Okay, or germ pool collection. Okay, so it's exactly the same thing, but just one more term added to it. Okay, now in your germplasm selection also, see, we are going to be conserving your plant, or we are going to try to bring something. So what we generally see is in germplasm, this thing. Now this is a little off related, but then I'll just let you know. Okay, when we talk about germplasm conservation, now what is happening is, because of this, like because of like excessive cutting down of forest, deforestation and all, sometimes there is a possibility we are losing particular parts of the plant, meaning particular characteristics of plants, or maybe we are like losing like a plant with good characteristics and all. So what we need to do is we need to save these plants, because we need to save them for the next generations and the near future as well. So what we do in this case is we go for conservation of such plants. Okay, so germplasm conservation. Now, guys, how can this conservation be brought about? Okay, once again, I'm telling you guys, this is not exactly related to what we are doing, but then this is a part out over there, small part. Now, guys, see, let's say we are having loads and loads of your mango trees, okay? And these mango trees, like, they are like, really important. But suddenly something happens. And guys, recently, did you know that, there is this one person who sowed some mango plant, and then, it is giving him like the costliest mangoes ever. He has kept 12 dogs and some guards to protect it, right? That is kind of what we are trying to say. Okay, that is nothing but we are trying to conserve it. How will you conserve it? Now there are two ways of conservation. Either it can be in situ in situ conservation, or it can be ex situ conservation. In situ or ex situ conservation. Now what is the difference between the two? Now guys, what you will do is in situ conservation, you are going to preserve it in the same place. Now wherever it is present at that place, so the plants are preserved in their original location. And how can this be achieved? This can be achieved by converting that area, like wherever you are going to see this plants, into your either natural reserves, or you can just convert it into your forest, or you can just announce this as forest, where all the illegal activities, like or like cutting down of plants for timber or their plant products, that is going to be completely stopped. Okay, or you can convert it into a national park. Okay, there is a little bit difference between what is a natural reserve and a national park. Remember, in national parks, okay, what you're going to see, a little bit of activity is still allowed, meaning some people who are staying in the national park, they can go and they can remove fruits from the plants, they can remove a little bit of wood from the plant. That all is still allowed in your national park. But in your natural reserves and forest, this is completely prohibited. Okay, this is the difference, guys. So here, when we talk about your in situ conservation, the plants, they are going to be preserved right in their place only. They don't have to travel anywhere. They are protected in that environment. But in your ex situ, ex situ means you are going to remove the plant from its place, and these are now going to be conserved. The plants are going to be conserved in either botanical gardens, okay, or you can have like some kind of your seed banks, or you can have some nurseries where these plants, they are going to be they are going to be stored, and then as and when required, they are going to be dispatched from these places. So guys, see, there are two different methods of preservation. Can you tell me, naturally, what is going to be better, in situ or ex situ? Naturally, I'm saying, no? Yes, guys, what do you think? Very nice, very nice. Naturally, it is going to be in situ only, because in ex situ you have to have you have to overcome some kind of hurdles. Like for example, one plant may not be suitable for the botanical garden in some another place, part of the country. The environmental conditions may differ, climatic conditions may differ, the type of soil may differ, the nutrients may differ. So there are many things that are going to change in your ex situ conservation. So always better, naturally, we'll try to always save them in situ. Okay, so very nice. Those who have answered, very nice. Okay, now guys, now let's try to see this thing. Okay, let's try to move ahead and let's try to see. So guys, here, first thing, what have we done? Sorry, किधर गया? हाँ. Okay, what have we done out over here? See, here we have had an entire collection of your variability, meaning we have collected all the variable genes and we need to combine these genes. Okay, so now let's say, okay, once again taking back the example of a rose only. Okay, we have, let's say, like nicely fragrant rose. Okay, there is one plant with fragrant rose and it's short lifespan. Okay, so let's say fragrant rose with short lifespan, and then there is another one which has, small roses. Okay, small roses or not so fragrant roses, but with large lifespan. Okay, now what is our aim going to be? Okay, our aim is to combine these two characteristics. Okay, so let's imagine we have to combine these two characteristics, but these are our plants that we have. So the second step that we are going to go for is we are going to go for evaluation and selection of parents. Okay, evaluation and selection of parents. Now guys, what are we going to do in this case? Now see, just like in your animal breeding we had selected who is a superior male, who is a superior female, right? That is what we are going to do out over here exactly. Now but now we are going to look for our desirable characteristics. Okay, so what are we going to do? Whatever germplasm that we had, okay, that germplasm is screened. Okay, it is going to be screened. You are going to look out which characters you want to you want to like go for. Okay, so germplasm is seen and the characters are selected. Okay, 1 minute, there's one question out over here. Sir, here this we are talking about the steps. Yes, yes, we are all talking about the steps. See here, see steps for hybridization. First, collection of your variability. Second, evaluation and screening or selection of your parents. Okay, now, okay, so we are actually selecting your parents. But now guys, once again, when we talk about the parents, how should the parents be, guys? The parents should be, they must be healthy. They should be healthy, they must be vigorous, and they should show complementary features. Hey, they should show complementary features, because then only we can go for this. नहीं तो एक feature अलग दूसरा feature अलग, we cannot go. See, complementary features meaning fragrant rose, small rose but with not so fragrant, so non-fragrant, I just put down non-fragrant small rose. Okay, non-fragrant small rose and short lifespan, large fine. So you are going to select parents in such a way that they are complementary to one another. Need to either fragrant rose, and here you will select that, a flower with thorns, or your short lifespan, and the other character you will select is like, morphological characteristics. Then it won't make sense. Then you cannot do a cross between these two. Cross has to be done by selecting these complementary characters only. Then only it is going to happen. But now guys, see, now once you have selected, you have made up your parents. Okay, so now you have, after screening this thing, you are going to select whatever your parents are. Okay. But now, guys, I need to know whether these parents are pure or no. So, in order to understand whether they are going to be pure, so these parents, whatever you have selected, they are going to undergo self pollination. And once again, for how long? Self pollination for a period of minimum 3 to 4 generations. Okay, 3 to 4 generation, so as to ensure that they are pure plants only. Therefore, it is ensured they are, and if the characteristics are true, okay, to ensure that they are pure parents only. Okay, that is what we need to do. Okay, so guys, once we have got this pure parents, then what you can do, then you can take the seeds and you can keep on sowing them everywhere, 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 and get multiple copies of this parent. Okay, fine. Chalo. So now we have got this thing. Next thing is what? Next is the most important thing, that is our actual hybridization. So now what are, we have got, two plants. Okay, sorry, third thing. Okay, so see, now we have got our two parents, okay, two pure parents. Now what are we going to do? We are going to bring about crossing. Okay, so now we are going to see exactly the same thing, like crossing over, like how we did it in Mendel's case. That is what we are going to do. Okay, so we are going to have, one is who's going to be the female, one is going to be the male. So in hybridization, one parent is going to be your male parent, and the other one is going to be the female parent. Okay, but now let's say, okay, we are talking about the male parent. The male parent, let's say he had, like good character. He was the one, maybe, like a good character, or the one without this thing. So guys, we have to compare. I mean, we have to mix their characteristics. That is what we are going to do. So for this, what are we going to go for, guys? We are going to go for your cross pollination. So both of them, they are going to be cross pollinated. And after your cross pollination, what are we going to get, guys? We are going to get the F1 generation. But now the F1 generation that is going to be there, this is going to be a hybrid of in both the characters. Like, now this is going to be what? It is going to be fragrant rose and large lifespan. So that way. So the F1 offspring, this is going to be hybrid for both characters. Okay, but now once again, you have to screen this thing, because see, I told you other way around is also possible. So you have to screen this thing. This is going to be shit. Okay, this is going to be screened, and finally the F1 hybrid is going to be F1 hybrid with desired combination is selected. Okay, fine. So this is what is happening. Okay, so what have we got? Now we have got our F1 hybrid with our desired characteristic, meaning nice and fragrant flowers and large lifespan. But now our job is not yet done. Our job is not yet done. Why? Because we still need to find out whether it is going to be the same thing or no. Like, whether this hybrid is really true or whether this is false hybrid. So once again, we have to go for the selection only. Okay, so let's try to see what is going to happen. So from these hybrids that we have got, okay, we are going to go for your selection and testing of superior recombinant. Okay, now what are we going to do, guys? Once again, see, we have got the superior ones out over here. Say we had selected this hybrid, right? The one with this thing, but we need to know whether this is going to be pure or no. So now what? We have selected the hybrid, the desired hybrid, and then how are you going to know whether this is going to be pure, whether this is impure? Once again, you are going to bring about your self pollination. Okay, so once again, as you bring about self pollination for a few generations, you are immediately going to know whether this plant is a pure for that character or whether it was your heterozygous. Okay, so once again, immediately you come to know that this is going to be your pure plant. Fine. Okay, now we have got our pure plant. Okay, the thing that we were desiring for, but still our job is not yet over. The reason is after this, we need to test this thing, because see, testing is very important out over here, because we just cannot, maybe this plant, in the first attempt, like, meaning in its external morphological characteristic, it seems to be fine. But now this plant may have some kind of your hidden mutations. It may have some kind of your drawbacks inside of it, which need to be evaluated, right? Otherwise what will happen is, you take this plant, it is going to have defective genome, okay, defective genes, defective DNA, and you have just put it into the wild. And then what happens? Immediately you will see that, oh, the bees and all, they are going to take the pollen grains from here, but those pollen grains are going to be defective. And when they are going to pollinate actual plants, they are going to contaminate the gene pool. They are going to, like, they are going to bring about even more worse mutations and even worse products. So that is what we don't want. So for this selection, after selection, testing of these is going to be very, very important. So the next step is testing, release, and commercialization of cultivars. Testing, release, and commercialization of new cultivars. Now, what is cultivar? Cultivar is nothing but cultural variety. Okay, so the one that you are going to get is nothing. This pure plant, this is nothing but your cultivar. Okay, so now what is going to happen is this pure parent, whatever we had again, the pure parent or the pure line, whatever we had got, this pure parent, this is now going to be evaluated. Okay, so how is this going to be evaluated? It is going to be evaluated for disease resistance. Okay, it can be disease resistance, or it can be your pest resistance. Okay, or it can be whether it can be, superior quality, like, increased yield, or whether this is like, ability to grow in harsh conditions, etc. Okay, or maybe, increased uh, nutrition value, etc, etc, etc. Okay, so we are going to look for all this thing. But now what is going to happen? See, guys, so immediately once you have got a new variety, you need to test this thing and you need to get it approved. Unless and until you don't approve it, you cannot release it into the market. So now what happens is, now you're going to say Indian Council of Agricultural Research. This is going to come into action. Okay, so this is Indian Council of Agricultural Research. Okay, this is like a very important body. Okay, ICAR. Okay, now what happens is, guys, once you have got this new variety, you have to submit it to ICAR. Now, what is this ICAR going to do? First, it is going to grow them into your control conditions. Okay, so first they are grown in controlled conditions to see whether the characteristics are true or no. Okay, they are all going to be first grown under controlled conditions. Controlled conditions of what? Controlled conditions of your water. Okay, it is going to be your temperature, it is going to be your fertilizers, it is going to be your aeration. Okay, aeration, etc. Okay, so all these characters are first going to be seen. Okay, now when they grow this thing, okay, in your controlled environment, in your laboratory kind of environment, if the plant is showing the same characteristics over and over again, okay, then it is going to be fine. Okay, then it is like, they are like, okay, so this thing is this thing has passed the initial test. But now once they have passed the initial test, I still have to go for the next thing. Now what is going to happen, guys, after this? Secondly, okay, what are they going to do? Second, they are now going to plant this thing for a minimum of 3 to 4 generations. Okay, absolutely very important. Okay, these are then grown in natural fields. They are then grown in natural fields for a minimum period of 3 generations, 3 or 4 generations, for minimum 3 generations. Okay, and now if you are like like if you are like past this test, okay, then what is going to happen, guys? First of all, three generations in natural fields, plus you are also going to grow this thing in different climatic conditions. Okay, so all this thing has to be done. And now once this thing is done, and in different climatic conditions, now once this that the plant has passed all these things and it is still going to give you the great results, then what, finally this is going to be released as a new variety, okay, by Iker is going to give you a certificate stating that yes, you have come up with a new variety, and then you can use it. Okay, so third thing that is going to be, if everything is working fine, then finally released as new variety, that way. Okay, so this is how it is going to happen, guys. Okay, and with this, by this method, guys, we like this guy Norman E. Borlaug, he had come up with this green revolution, and because of this green revolution, he has saved so many developing and underdeveloped countries. Okay, but what did he do? How it is? We'll see it in a while. Okay, but before that, guys, once again, just see if everybody is okay with this. Guys, this question will be asked for 4 marks for you guys. Earlier, it used to be asked for 7 marks. Okay, so what are the main steps, guys? Okay, you don't even have to remember, you don't even have to mug this thing up. Just think about Mendel's experiment. What did you do for Mendel experiment? First of all, you selected some characteristics from a group of characteristics, so that is nothing but collection of germplasm. Second thing, you isolated your pure parents, so you know the characteristic which you wanted, so second step is that. Third thing, you brought about cross pollination. Here we are just calling this as hybridization. So you bring about cross pollination, and then what, you are going to get your F1 hybrid. Now from those F1 hybrids, you are going to see which is that characteristic that you're looking for. So next is your screening. Okay, so screening and selection of the superior recombinant. Once that thing is done, we need to be still sure that you are going to undergo like self pollination for a minimum period of 3 generations to make sure that the characteristics remain true. Okay, and now what happens after your the next step, then once you are sure that, oh yes, this plant is a new variety, it has combined characteristics, then you are going to just go to Iker, you're going to submit your claim. The Iker is going to first grow it into your controlled environment, where you will see that yes, if it is working properly, then it is going to be grown in your natural fields. Once it is grown into natural fields, if it is still performing the best, then immediately this is going to be declared as a new variety. Okay, everybody all right with this thing, guys? Sure, no problem. Understood this part, guys? You just need to connect this with your Mendel's experiment. How are a neat question? How are questions asked from this in neat? So then they will ask you, which committee is responsible for testing of new cultivars? So that is going to be ICAR. Okay, or they may ask you like, in order to continue with the testing and release, you need to ensure that which hybrid is going to be selected. So it is going to be a recombinant with both the important characteristics. So something of that sort. So from this part, the questions are going to be really, really simple and straightforward. There's no much twisting and turning. But then the next part of the chapter is going to have all the hideous questions, because you have loads and loads and loads of examples out over there. That is where things go a little tricky. Okay, so okay, but now guys, what we'll do, I'll just give you a quick 5 minute break. Okay, we are just about 8:30 right now. Okay, anyways, 5 minutes break, guys. Anybody who wants to go to the loo and have water, go and come quickly, guys. Yes, 5 minutes. 5 minutes, quickly. Yes, yes, Himanshu. Yes, yes, Aryan, no issues. Definitely. Okay, chalo. So guys, once again, whatever I have just said out over here, like how it is going to work and all, okay, the same thing has been given in your textbook as well. Okay, like these points are also there, but just to keep in mind, like you can keep this thing. Okay, this is the steps that are there which we need to follow. Okay, it's exactly the same thing, nothing different as such. Okay, so just have a look. Whatever I just said right now, na guys, it's exactly the same thing. Okay, chalo. So guys, let's try to move ahead and let's try to see the next thing, guys. Okay. So guys, the next thing that we need to talk about is what is this green revolution? Okay, now what happened was, uh, in 1960s, okay, somewhere in the 1960s and all, the world was like, really, really having great depression in the production of your wheat and rice, and especially in countries like India, Mexico, the Middle East and all, we were like really struggling with the normal required daily requirement, like to fulfill the daily requirement of the people. So now what happened was, there had to be like some or the other way by which we could increase production, and that production was brought about by using this technology, this plant hybridization technology. And this was given by a person whose name was Norman E. Borlaug. Okay, and because he brought about this kind of revolution, guys, remember this revolution is also known as Green Revolution. Okay, guys, there is another revolution which is called as White Revolution. Can anybody tell me what is White Revolution? Anybody, if anybody knows White Revolution, anyone? Is anybody aware of White Revolution? Yes, very nice, Himanshu. Very nice, Sneha Singh. Yes, it is going to be for your milk production. Very nice, Varghese Korean. Very nice, Sneha. Yes, very nice. Okay, so basically, it is for your milk production. Same way, this is going to be green revolution. It is for your food production, especially your wheat and rice varieties. And guys, this technique was so nice. It was so proficient. That you know immediately the production of rice and wheat, it just increased manifold. It increased exponentially, and immediately all these countries where this kind of plan was like initiated and it was executed, that entire those countries, they became self-sufficient in production of their food reserves that way. So guys, this person who is responsible for this, his name is Dr. Norman E. Borlaug. Okay, and this guy, he is known as father of green revolution. Okay, so basically, when we talk about this person, what did he do? He brought some experiments on your wheat. Okay, what were his experiments? We'll talk about. But guys, just like we have Norman E. Borlaug, same way, guys, we have another person who initiated the same thing in India, and he is Dr. M.S. Swaminathan. Okay, and guys, this is a very, very important question from the NEET point of view. Always asked. Okay, the person to initiate uh, this thing, Green Revolution in India, he is M.S. Swaminathan, and this guy, he is known as father of Green Revolution in India. Okay, so first we just see, like, what did these people do and why do we really know them for? So guys, when we talk about your Norman E. Borlaug, this guy, he was working on semi-dwarf varieties of wheat. Okay, now these semi-dwarf varieties, they did not take much space also, and plus they were like growing really rapidly. That is why this was important. Semi-dwarf varieties of wheat and rice. Okay, but now when we talk about this Dr. M.S. Swaminathan, what did he do? He also worked on the same kind of principles, okay, but he was also involved in your mutation breeding. Okay, so same principles, same thing, like, he also worked on basically your wheat only, the same thing, but he is more and known for mutation breeding. We will see a little ahead what is this mutation breeding. It is basically by the use of using mutagens. Okay, so it can be your UV mutagen, it can be your chemical mutagen. He tried to bring about mutations, and he tried to say, like, whether it is going to help in the production, so that way. Okay, but then guys, what did these people develop? So let's try to see. So when we talk about Norman E. Borlaug, basically I said he is going to work on different kinds of your rice and wheat. But what are the different types of rice and wheat that he has given? So first of all, we will talk about what are the wheat varieties. Okay, and then we'll talk about what are the rice varieties. Okay, so the wheat varieties, guys, and guys, this is the most important this thing. Okay, okay, so what are the wheat varieties that he had come up with? So we have Kalyan Sona. Okay, first one, Kalyan Sona, Sonalika. Kalyan Sona, Sonalika. Then we have Sharbati Sonora. Okay, so we have Sharbati Sonora. Then we have Sonora, normal, just Sonora 64. And then we have your Lerma Rojo. Okay, so we have Lerma Rojo, that is going to be 64 once again. And the last one we have is Lerma Safed. Okay, these are the plants that were developed out over there. Okay, so you need to remember this, okay, guys. And the next one is we have the rice varieties. Now, what were the rice varieties out over there? Okay, so we have first Padma. We have Padma Kanti. Okay, then we have IR8. Okay, now just remember this thing. IR8 is it is basically comes from your Indian research, agricultural research thing. Okay, so IR8, ED. Then we have your Taichung native. Okay, Taichung native one. Okay, fine. Then we have your Ratna Jaya. Okay, Ratna Jaya, and finally we are going to have your Vijaya and Jayanti. Vijaya and Jayanti. Oh my god. Now, the problem is, guys, you need to remember this. Okay, and how are you going to remember this? Okay, let's imagine you are in a market, okay, and there is a person who is selling tortoise. Okay, and he is calling out to you, and he is kaso saso lele, kaso saso lele. Just remember this thing as kaso saso. Okay, there are like the rhyming terms. Kaso saso lele. Okay, doesn't make sense, but it will help you to remember. Okay. हाँ, Jaya Vijayanti सबकी पसंद निर्मा. हाँ. Okay, yes. Okay, and you can remember this thing. Okay, you can just remember this thing. Pick it, pick it, RJ VJ. I remember it this way. Pick it, RJ VJ. Okay, so Padma Kanti, IR8, Taichung native, Ratna Jaya Vijaya Jayanti. That way. Okay, so guys, either you follow this thing, pick it RJVJ, or you follow Kasho Saso Lele, or you make your own things. Okay, but you have to know it this way only, guys. Okay, you need to know this because in your NEET exam, guys, just imagine there are going to be some more 25, 30 names out over here in this chapter. You need to remember who is who. I don't want to waste your memory space. Okay, make this short acronyms, stick to it, and whenever the question comes, just go back to the acronym, see whether it is out over there or no, and just write the answer. Don't mug this thing up, guys. Don't mug this thing up, because this is really going to be confusing later on. Okay, fine. Next, so but now when we talk about this guy, okay, Swaminathan, Dr. MS Swaminathan, what did he do? So basically, some of his works, it indicates that he produced some wheat, and wheat like what? Wheat like Sonora. Okay, so one is Sorona, then second one he had come up with Sarbati, Sonora, Sarbati, and last one it was your NP 165. Okay, so something like this. So just remember this, you can always remember this is small. Okay, so this is what we need to know with regards to your Norman E. Borlaug and your MS Swaminathan. So just have a look, guys. If everybody is okay, then we can move ahead with the next part. Yes, yes, yes. These are all, they're part of the same program. Padma, Kanti, Ratna, Jaya, Vijaya, Jayanti, they have all been developed in India only. That is why these names. Taichung native is from Japan, I believe. Uh, Among these, which is the most eaten one wheat? No, 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 no. We have we eat all of them. So some places will have more of Sharbati Sonora. Some of them will have Lerma Safed. So we eat like in different places. We have different things. See, if you ever go to a shop, okay, like if I'll just tell you, if you have ever gone to a grocery shop, you will see that there are different types of rice. Okay, koi hai kolam. NP 165 is the last word I've written. NP. Okay, see, there is kolam, then there is basmati, then there is this, then there is that. I say tukda tandul, ye tandul, wo tandul, right? All of these are what? These are nothing but different varieties, and all of them, they are in demand because every person has a different preference. So, for example, like for your biryani, okay, let's say you are going to make biryani. So for that, I don't think so. You are going to use normal rice for it. Rather, what are you going to use? You are going to use basmati rice, right? So the same way, every region is going to have their own likings. So that way, so no such specific thing. We have developed various varieties, and all of these are yes, Odisha people is going to prefer sticky rice. If you go down south Kerala, you are going to see they will have more of the brown rice and the sticky rice once again. Okay, so those rice now, if you have ever seen South Indian rices, basically, okay, there may be a few South Indian people out over here who may be well versed with it. Okay, those are big grains. Okay, if you look at the Maharashtrian peoples, they have like small grains, एकदम ऐसा full थोड़ा सा सुकड़ा बोम्बिल जैसा, and those have a little big ones, and they are going to be a little fluffy types. So when you cook them, also it becomes like a nice and fluffy. Huh. Yes. Right. Okay. Then there is one more question. I have heard all the names. One matter. Norman. Norman was from where? He was from America. He was American. Okay, and basically later on, he also got this Nobel Prize, huh, for this thing. Okay, chalo. So guys, let's try to move on. Move on with the next one. So see, just like how it happened with your wheat and rice. The same thing people try to do with your this thing as well, with your sugar canes. Okay, so let's try to see with regards to sugarcane how it is going to be and what really happened out over there. Okay, so first was your wheat and rice. Second one, we have sugarcane. Let's try to understand what are we going to see with sugarcane. Okay, so basically, especially in India, what happens is we are going to have two types of sugarcane. Okay, there are two types. One is Saccharum barberi and one is Saccharum officinarum. Okay, sure. So we have Saccharum barberi and the second one is Saccharum officinarum. Okay, now guys, when we talk about these sugar canes, okay, both of them they have two different targets altogether. Okay, now how it is going to be? So when we talk about this Saccharum barberi, guys, Saccharum barberi is the one which is going to grow in the north. Okay, so this is the one which is generally grown or cultivated in the north. Whereas when we talk about this Saccharum officinarum, this is the one which grows especially in the south. Okay, but now you know what, guys? Both of them they have different contents. Okay, they have different contents altogether. Okay, let's try to understand what it is going to be. Okay, so first thing is when we talk about this Saccharum barberi. Okay, Saccharum barberi is the one who is going to have like not so good yields as such. Okay, so not so good yield. Okay, it is like average. Okay, not so good yield. Second thing, this also has less sugar content. Okay, not so good yield. Plus, second, it has less sugar content. But then, when we talk about this, when it grows in the north, it is going to grow like really, really well. So, grows well in the north. Okay, but now the problem is, okay, when we compare it with officinarum, there are a little changes. But then one more thing, this is going to be highly resistant to your diseases as well. Okay, so it has some good qualities and it also has some bad qualities. Okay, now what happens? Okay, when we compare this thing with our Saccharum officinarum, this one it gives good yields. Okay, so yes, so here it has very very nice yields. Second thing, guys, this had a thicker stem. Okay, so it's like really really nice and juicy. Okay, thicker stem, and plus because it is nice and juicy thicker stem, it has high sugar content, which is especially what we are really looking for. Okay, when we talk about a sugar cane, so high sugar content. Okay, but the problem is this does not grow well in the north, and that is why it is going to be grown only and only in the south. Okay, it doesn't grow well in the north. Okay, and plus there is no information about whether it is disease resistant or no. Now what people did was people said that, oh, we'll follow the same principles, we'll follow the principles of your hybridization, and then let's try to see what can be done. So what they did was they took both of them, Saccharum barberi and Saccharum officinarum, and one thing that they did was they brought about cross pollination. Okay. And now, after cross pollination, what are we going to get? We are going to get hybridization. And by hybridization, guys, once again, we are going to get your new offspring. So we get this F1 hybrid. And this is the one. Guys, now what happened? We started getting different kinds of hybrids with good qualities of both. For example, we got some of them like Coimbatore. Okay, now CO is Coimbatore. Coimbatore 419, 421, 453. These are really, really nice varieties. Okay, now Okay, these are some of the hybrids that we got. Now guys, is it like important to know these numbers? The answer is no. Okay, you can just write down some random numbers out over here, even if it is mismatching, that is still fine. Just don't put down Coimbatore 420. Okay, don't put it Coimbatore 111, Coimbatore 666. Okay, don't put such kind of numbers, put some random numbers always. Okay, however, these are like real plants. It is not that these are like fake plants, these are like real plants. Okay, and all of them, they were developed at an institute in Coimbatore. Okay. So, guys, that's it. That is what we needed to know with regards to your sugarcane. Okay, Saccharum barberi and Saccharum officinarum. The same kind of thing, guys, we have also done it for your millets. Okay, millets are what? These are like, ragi. Okay, these are like, chota chota dana types. Okay, those are especially called as your millets. And once again, okay, like from the millet family, who are we going to have? We are going to have maize, jawar, bajra. Okay, those are also going to be from the millet family. So, guys, how we have gone ahead, we have done the same kind of hybridization things, and we have produced like hybrid maize. Guys, remember the names are very important. Okay, hybrid maize Ganga 3. We have Jawar, hybrid Jawar, Coimbatore. Once again, Coimbatore 12. Okay, we have Bajra. Okay, all these things they have been done. And now, guys, whenever we did this thing, now, guys, once again, very important, you have to remember these are high yielding varieties that is there. But plus, you are going to see that these are water stress resistant. They can still survive in less amounts of water. Okay, so this is what is going to be important. That's it, guys. Why are these things important? You need to remember this because these are going to be asked in your NEET exams rather than straightforward questions from like the first part. They generally tend to ask you these examples, or they will give you like 5 examples of your rice and 1 example of wheat, and they'll ask you to find out who is the odd man out. Okay, so you need to know these examples properly. Okay, everybody all right with this thing? Just have a look, guys, if you're okay with this. Okay, so with this we are done with like the first part, like like hybrid crops and all. The second thing we have to talk about is like how are we going to plant how are you going to breed plants for disease resistance, and then we have like some specific plants developed for your different kinds of your resistances. So that is what we are going to do in the next class, guys. Okay, next class hopefully we should be in a good position to finish this thing off. Yeah, almost. Okay, so guys, everybody all right with this thing? Sure, no issues. Everybody okay? Good, guys? Okay, chalo. So guys, once again, we'll stop over here for the day. Only Aryan, okay, who wanted to speak something, okay, you can stay back. Rest everyone else can leave. Okay, bye bye. Bye everybody. Bye bye. Acha, okay, okay, no problem. Aryan, call me. Yeah, okay, okay, yeah. Okay, bye bye. Bye. Bye everybody. Bye bye.